We can start now. My name is Simar Rapadre. Today I'll be doing a workshop on heredity and genetics. Just a little bit of rules and disclaimers before we start off the workshop. Please do not be disruptive in any way possible. Don't Please be courteous to the instructor and any TAs helping, and have fun. If you are disruptive, you'll have to remove you from the workshop. That's to help the other students that are actually listening and taking notes to help them better concentrate. So yeah. So we can start off. What is heredity exactly? The cookie cutter definition of heredity is it's the study of inheritance. Children inherit traits from their parents, and these traits are passed on throughout the family generations. And these traits aren't just linked to physical or internal changes. Either there are a few traits that may not even seem like traits. Here are some examples of them. Baldness is one of them. Uh, while Okay, okay. So while the gene for baldness is found on the X chromosome, that men only get from their mother's side. Uh, yes, you have a question? Okay,
Not really. I just wanted to also share that they also had you can also another heredity based thing is autism. If your parent has autism, you'd have twice. I heard somewhere that if your parent had autism, you'd have some amount higher chance of getting autism yourself.
hmm. Yeah, that would make sense. It's based on the genes that you have. If one of your parents has autism, you have a higher chance of developing autism. So yeah, that's a good one. It's a good one. Um, Moving on with the baldness side, intelligence is also a slight trait that you can get, and it mainly comes from your mother. The genes that determine intelligence are located in the X chromosome, and since women carry two X chromosomes and men only carry one, children are twice as likely to inherit their intelligence from their mother, so their base intelligence. And also another one that doesn't really matter is drinking black coffee. It's inherited from your parents, but the environment can also play a big role in that. So, what even happened? Oops. All right, I don't know what happened there. So the location of these genes, as most of you know, DNA and genes are located in cells, which are the basic functional unit of all living organisms. It's a little bit about cells. They're made up of every living thing is made up of one or more cells. All living cells are come from pre-existing cells, and they are formed via cell division. And the cell is the basic unit of structure and function in an organism. DNA and chromosomes. Chromosomes are made of DNA. They are condensed packages of deoxyribonucleic acid, and these DNA molecules contain several genes along its coding sequences. DNA itself is a double helix shaped molecule with a sugar phosphate backbone, and the way it codes for genes is that they have a nucleotide sequence of four nucleotides: adenine, guanine, cytosine, thymine, each represented by letters A, G, C, and T respectively. And the order in which these nucleotides are sequenced within the DNA molecule is is coded for a gene through a process called transcription and later translation into a protein, which helps display a trait. Genes are sections of DNA that contain hereditary information, and these are again instructions to make proteins in the human body, which display the traits. All right, so a little bit about inheritance. When we're born, we have two copies of each gene containing chromosomes. So we have one for our mother, one for our father. This should be pretty basic stuff. Each comes from a mother's egg or a father's sperm. The inherited traits are characteristics that are controlled by the genes and DNA passed down from parents to their offspring, and these include the very basic examples are height, your natural hair color, eye color, skin color, the length of your fingers, uh, the amount of hair you have on your body. All that just comes from your parents. Alleles are variations of the same genes. These are what causes the genetic differences in individuals. So each person is born with two alleles or two versions of the same gene. They can be two identical alleles or two different alleles, as you can see in the example here. Eye color has several alleles: brown, blue, green, gray. There's some more, but these are the four most common ones, as well as hair color: blondes, redheads, brown haired, and black haired. And these alleles interact with each other to produce a phenotype or a displayed trait. So with alleles, there will be a dominant allele and a recessive allele present, at least one of each present in a whole gene pool. If an individual has a dominant allele, then the dominant trait or the trait that the dominant allele expresses will always be expressed, even if the second allele is recessive. But if an individual has two recessive alleles, then the recessive trait will be expressed. And these alleles are represented on a Punnett square or dihybrid cross, or any other inheriting figure by an uppercase letter symbolizing the dominant allele and a recessive letter symbolizing the recessive allele, or lowercase letter. And here is an example of an inheritance tree with purple as dominant. So you can see purple, the dominant is the dominant. This plant is homozygous dominant for purple because all of the F1 offspring are purple, but these are all heterozygous since the parent is homozygous recessive for the white color. If you don't know what those terms homozygous or heterozygous mean, we'll get onto it in a few slides. And when these organisms are crossed, 3/4 of the plants have purple flowers and 1/4 have white flowers due to the crossbred nature between the purple and white flowers. And here's the example of a Punnett square. So you can see here two heterozygous or mixed crossbred individuals are bred, and their eye color is heterozygous. So the offspring will be 3 brown-eyed and 1 blue-eyed. So when the parents are heterozygous, unless it's on the sex-linked chromosome, which we'll get into later, the organisms will always have 3 dominant phenotypes and 1 recessive phenotypes. And within this cross, we can see that one of these 3 dominant phenotypes is fully homozygous dominant for the allele or for the phenotype, and 2 are heterozygous. And again, here it's a similar thing, heterozygous and homozygous recessive this time. So here there's going to be 2 heterozygous individuals and 2 homozygous recessive individuals. Here you can see the key with the dominant recessive alleles and the phenotype matching up with the genotype. So more about genotypes and phenotypes in of itself. When we refer to an individual being as homozygous for a certain gene, we refer to them as having 2 copies of the same allele, whether it be dominant allele or recessive allele, which is homozygous dominant and homozygous recessive respectively. Heterozygous, on the other hand, refers to an individual who is crossbred for a certain gene. They have 1 dominant and 1 recessive allele. The homozygous or heterozygous nature of one's genes are what's called a phenotype, and the physical, or what's called a genotype, and the physical expression of these genes is what's called a phenotype. By the way, if you're stuck on any of these concepts or don't fully understand, you can unmute yourself and ask any questions at any time. And so this is a picture of a dihybrid cross, which is a much more complex version of a Punnett square. Here you can see it codes for this dihybrid cross displays 2 different alleles at once. For one is for the color of the seed, and one is for the texture, whether it be round or wrinkled. And by putting it in a dihybrid cross, you can see the likelihood of both the alleles showing up in one different organism. And you can see since both of these organisms are homozygous, or not homozygous, both these organisms are heterozygous for both of the alleles or both of the genes, you can see that 9 out of 16 will be homozygous dominant for both. 3 out of 16 for each of them will be homozygous and heterozygous, I believe. And then one of them will be completely homozygous recessive. And here's some other types of inheritance. So not everything follows the dominant recessive allele figure. Some genes are incompletely dominant. So as the name would imply, one allele is not completely dominant over the other, and as a result of this, the alleles blend together and cause a new trait to appear. So an example of this would be a red flower crossbred with a white flower, which produce pink flowers as a result of the red and white mixing in. On the other hand, you have codominance. It's when both alleles are expressed cooperatively together in an equal fashion. And an example of this would be a spotted chicken arising from a black feathered mother and a white feathered father. So it would have black and white spots all over its body. Um, 1 second. All right. Here we get into genetic disorders. So a type of disorder here would be an autosomal disorder, which would have single genes and multiple genes out of scope. So autosomals can affect one single gene or multiple genes at the same time on a single chromosome. And you have sex linked, which is this similar thing. The disorders affect genes, but instead of being on normal chromosomes, they'd be on second chromosomes and chromosomal abnormalities. So again, autosomal disorders, they're carried on autosomal or non-sex chromosomes. An example would be Huntington's disease would be an autosomal dominant example. It would need only one dominant allele to have the disorder, and the autosomal recessive disorders like sickle cell anemia and cystic fibrosis need both recessive alleles to have the disorder. So Huntington's disease, it would affect the brain. Sickle cell anemia and cystic fibrosis are much less severe. Autosomal dominant diseases tend to be a lot more rare than autosomal recessive, but both are still disorders nonetheless. And then you have sex-linked diseases. And these are caused by errors in the genes of traits of the sex chromosomes, and they are still dominant or recessive, but work slightly different when looking at a cross. And since sex-linked alleles always show up in males, whether dominant or recessive, because males only have one X chromosome. So sex-linked alleles carried on the X chromosome. If a male inherits an X chromosome with a sex-linked disorder, then they will always express it no matter what, because they only have one X chromosome and they cannot. And the Y chromosome doesn't carry the same genes as the X chromosome. And an example of sex-linked diseases would be hemophilia and color blindness. Oh, so we're gonna pause here, and I'm gonna give a couple questions to you guys. I'll put them in the chat, and I'll give you a few minutes to answer them to try to solve the problems when you're done. So put them in the chat here. Are the X and Y chromosomes codominant? No, they are two completely separate chromosomes. A codominant chromosomes aren't codominant. The alleles are codominant. Since the X and Y chromosomes carry different genes, they can't be codominant for each other. That's a good question, though. Okay. Let me give one. All right. That is your prompt. I'll be asking a couple more questions to make sure you guys understood the material. So yeah, I'll give you around 3 to 5 minutes to answer this question. Maybe quicker if some of you already know the content. And if you don't know what carrier means that you have one copy of the gene, but it's not expressed. So in this case, the mother is heterozygous. Since hemophilia is recessive. It's a recessive allele. And she only has one copy of the recessive allele. She has a dominant copy of the other, so she's not afflicted by it. If you're stuck on how to solve it, a little hint would be to use a Punnett square, but you have to be careful. You can't. What? Why is it going through that? What? My bad. Still waiting on a couple more answers. One student has already given an answer for the question in chat. Okay, 3 people have submitted their answers. I'll wait a couple more minutes. Yeah, I'll tell you a solution for the problem. Wait for a little bit more. Wait for only 20 more seconds. Okay. So the correct answer was actually 25%. And although no one got it right, the actual solution for this problem is right here in the image. So I said that hemophilia was a recessive disorder, and the mother has 2 X chromosomes. So this is the tricky part of dealing with sex disorders. The mother has 2 X chromosomes, and the father only has an X and a Y. So if the father doesn't have hemophilia, he cannot carry the disease. So he's not heterozygous at all, which I saw some of you fall for. With the probability it could be heterozygous. So when these two have children, you're gonna have an unaffected son, an unaffected daughter, an affected son, and a carrier daughter. Because only one of because they got that this boys got the X chromosome for the mother, so none of the daughters can be affected. So they're gonna get their dominant allele from their father guaranteed, but the son have a 50% chance of being affected because of the recessive allele X linked there. So that's the tricky part of dealing with sex chromosomes. All right. We'll go to the next slide, then I'll ask another question. All right, so we have chromosomal abnormalities, and these can be caused during mistakes during meiosis. So if you don't know what meiosis is is sort of mitosis, or if you don't know what that is, meiosis is the division of sex cells. So it's the sperm or egg cells in a human body, and these can result in removal or addition of whole chromosomes, DNA segments, or genes. And here are some examples. So we have Down syndrome, which is an autosomal disorder. So it's on an autosomal chromosome. And the abnormality, it has a trisomy 21. So trisomy means that you have 3 copies of the same chromosome. Each person only has 2 copies of the chromosome, but in some cases they have 3. And Down syndrome, of course, results in slower mental development, slight physical deformities, and many other things. Edwards syndrome and Pateau syndrome are also very similar. So they have trisomies on 18 and on chromosome 18 and chromosome 13. Klinefelter syndrome is another sort of extra chromosome added, but it has an extra X chromosome, which brings the total count up from 46 to 47, and it happens on a sex chromosome, so it happens on the X chromosome. And Turner syndrome is sort of like the opposite. This happens in only females, and they have their second X chromosome removed. So they only have one X chromosome. And trisomy X is of course the exact opposite, where you have an extra X chromosome. And super male, this is when you have two Y chromosomes. Most individuals that have a super male syndrome don't really survive for that long. Maybe I believe they barely make it past the newborn stage. So yeah, they don't survive for too long. And you have other ones, which doesn't result in the whole additions or removals of chromosomes, but this is deletions of certain genes within the chromosome. How could someone inherit two Y chromosomes? When the sperm accidentally copies two Y chromosomes. So when the sperm has two Y chromosomes inside of it instead of one, through an error in meiosis. Yeah. So Creutzfeldt syndrome is an autosomal disorder that happens on chromosome 5, and this results to because of a partial deletion of the chromosome. So part of the chromosome is removed, and that messes with certain genes. It can remove whole genes or just cut off parts of the gene code to prevent coding of the gene. DiGeorge or velocardiofacial syndrome is a microdeletion of chromosome 22, gene 11. So this is where it chops off part of a certain gene and causes it to not function properly. And Prader-Willi syndrome is, again, is the deletion of a paternal chromosome 15. So when the father's chromosome 15 is deleted, and Angelman syndrome is when the mother's chromosome 15 is deleted. And here are some of the chromosomal abnormalities shown. You have the deletion or removal of part of the chromosome, duplication, which is the exact opposite. Inversion is when the gene code is flipped. So you have a gene code of, let me put it in chat. Say if you have a gene code of AGCTC, no, I'm not typing. AGCTCGAGCT. Say if that's the gene code. Inversion flips it on its head. So it would be, I believe, this. It would just reverse it on the same chromosome. Substitution is when part of a chromosome is translocated onto another chromosome. And translocation is, of course, so substitution is when it's translocated onto another chromosome. Like pure translocation is when parts of chromosomes are just full flipped. So substitution, you're not one chromosome is only giving, but translocation in both chromosomes are giving to each other. It's not related to crossing over at all. Translocation happens after meiosis and can result in probably the least amount of deficiency, but it's still pretty. It's still not a good thing. So yeah. Okay. Here is a little question for you guys. So this is going to be all the way back. So second, let me find one. All right, okay. So before we go all the way back, let me just ask you a little question about this slide. So that's the question. It's okay if you get this wrong. This is just based on your intuition or your opinion, or not your opinion. There's a definite answer to this, but it's just based on what you think after viewing the 5 types of chromosomal abnormalities and what they can all do. Uh, let's give you a couple more seconds to get your answers in. Only 3 people have answered so far. Okay. All right, that should be good enough. So you all had really good answers, but the correct answer was actually deletion. And the reason it's deletion is because not all of the chromosome or DNA actually codes for genes. With deletion, the most likely thing that will happen in the errors will be that the chromosome will delete a non-coding region of the DNA. With substitution or translocation or inversion, the non-coding region can actually become a coding region if translocated or substituted or inverted onto another gene, and can cause gene deficiency or an overexpression of a gene. Duplication is on the same level as deletion, but it's also more risky, slightly more risky, because if they duplicate both a non-coding region and a coding region, it can screw up the entire gene. But deletion has the least risk, if that makes sense. All right. So getting to some potential careers. Here's just some listed off. A genetic therapist or engineer is one who works with genes, works to suppress genes, and this also relates to CRISPR-Cas9. If you don't know what that is, it is a tool used by some scientists to sorry, alter genes directly, and this can be used to correct disorders. A doctor in, I believe, China used it on humans to separate conjoined twins, I believe, but um, it's not approved to use on humans yet, and the doctor was, I believe, arrested. Uh, and I don't think he's been sentenced yet, but he was arrested for doing it, although the experiment was a success. Uh, yes. Or
No,
were you just
you
putting your
actually
hand
just
down?
answered my question. I was just wondering if it works.
Oh, okay. If it works, yeah, it worked. It worked, but it wasn't fully approved. The twins could have easily just died. It was a huge risk to take. It worked, but the risk wasn't approved to be taken. If that makes sense. You could also be a microbiologist, someone who looks into specific parts of a cell or a human body at microbes, bacteria, and studies them and learns about them. And Silicon Valley, in particular, has a large booming medical field and biotech field, and the demand for scientists is growing by the day. Uh, there's a lot of different people that wasn't it Berkeley professor Jennifer who created CRISPR. I do not remember who created CRISPR. But if you're curious, you can look it up. It should be there. And just a little question to you guys: who here is interested in a field in genetics? Who is interested in a field such as a genetic therapist or a bioengineer? I know those two are pretty common for people coming out of this Silicon Valley area.
Well, it's sort of complicated for me because I'm interested in a lot of things. Like I love the idea of genetic scientists, of genetics and stuff, but I'm just less able to understand it, so probably less interested in like actually pursuing a career in the field.
Yeah, I understand. It's complicated, especially when you get into more complex things, such as again this CRISPR process and the actual genes themselves, as well as protein coding. And this is just the cookie cutter basics of heredity and genetics. It goes way deeper than this. And yeah. All right, got another person. Not necessarily genetics is biotech, medicine for sure. Okay, that's good. That's good. Medicine is a great field, and didn't any more people going into it? Working with CRISPR seems fine, interesting enough. Yeah. Okay. All right. So that is the end of the presentation. Does anyone have any questions?
No?
For a couple of minutes. Actually, while we're waiting here, I'll give you one last question. Is there a Kahoot? No, I don't believe so. I'll give you guys one last question here. If a woman inherits an X chromosome that carries an affected trait, and one doesn't carry an affected trait, how is it determined whether they would be an affected or a carrier? Okay, it depends on if the male has the affected trait too. So if the male that she has children with has the affected trait as well, then one of their daughters will be guaranteed to have an effect. They'll be guaranteed to have an affected daughter. Um, if they if the disease is that's only if the disease is autosomal recessive. If the disease is autosomal dominant, or not autosomal dominant, if the disease is X-linked dominant, then all their children will be affected. Or not all their children. Sorry, my bad. I believe half their children would be affected. If it's recessive though, then only one son will be affected. If the male does not have the disease, if he does have the disease, the daughter will be affected too. Uh, and if it's an X-linked dominant, well, both boys, yeah, or half the children will be affected because if the mother doesn't pass on if the mother has one dominant allele and one recessive and it's dominant, then only half her children will be affected. All right. So there's the last question for you guys. This might take a little bit of time if you don't remember the hybrid cross from earlier. If you do, then this should be easy. 25%. We have one person that turned in. 25% another person. I'll give you a little bit more time, maybe 2 more minutes. Maybe like 1 more minute. Okay. So if you can go back here to the dihybrid crosses initially, you can see the answer is 9 out of 16. Wait, am I stupid? Never mind. Oh yeah, never mind. Wait, okay, never mind. Yeah, okay, no, it's just one. It's just one. For homozygous dominant for both, it's gonna be just 1 out of 16. So yeah. 25% was close. But it didn't say homozygous dominant for, I believe, both of them, or for one of each. I asked for homozygous dominant for both together, and yeah, it's gonna be 1 out of 16. So 1 out of 4, 1 out of 4. Okay. So yeah, that's pretty much the end. Is there any more questions that you want to? Kind of like 1 out of 16 to mix the percents up. Yeah, it happens. It happens. It's fine. Are there any more
I had a
questions?
question about translocation.
Mhm.
Is there any examples of genetic diseases that use translocation?
Yeah, there should be some diseases. If I can think off the top of my head about one. Um, I remember. I'm remembering the effects of one, but uh, oh yeah, wait, no, I'm also stupid. Yeah, never mind. Down syndrome is a translocation one. It's where
Oh,
part of
okay.
one chromosome breaks off and starts another 21st chromosome.
I see.
So yeah. Are there any more questions? Right. So, will we get a recording of this class? Yes, we will. There will be a recording, I believe. Yeah. Okay. So if there are no more questions, then I believe you guys are free to go. Thank you for coming out to this workshop, and yeah, I hope you find some fun in learning about genetics.
Thank you for the class. I found it really enjoying. Bye.
Bye. Okay.